everyone, welcome back to the cell signaling course, and today we are going to learn something as a summary of what we have been seeing over multiple classes before this. We have seen that how receptors are activated, we have seen how ligands are regulated, how certain events lead to activation, inactivation of the effector molecule, how adapters are engaged and so on. But the signaling which is initiated with receptor activation must end at some point of time. That has to be decided, that has to be precisely controlled. So we will understand that how by different means receptors were regulated. But before that we will see how different ways receptors were activated. But in general, what we understand from any of these signaling which we have discussed, whether it was about a receptor which had internal domain, which had an enzymatic activity, and we discussed largely the enzymatic activity of kinase type, or there are receptors which do not have any activity, enzymatic activity associated with them, rather they associate with an enzyme. Or the ultimate class which we saw very recently of this GPCRs, where depending on the G alpha, which is the constant activity of GTPase, you recruit next molecule with some enzymatic activity, like we saw adenylyl cyclase, we saw phosphodiesterase, we saw PLC or phospholipase C activity. The end result of all these things, whether it involves only one intermediate, like in case of two-component system, simple canonical two-component system, or multiple intermediates which we saw with the GPCR, which we saw with receptor tyrosine kinases in animals, or we saw with receptor-like kinases in plants, as well as the hybrid two-component system. In all these cases, what exactly happened was that an effector molecule is activated. Whether the effector has a protein-protein interaction activity, or it has enzymatic activity, or it has a transcription factor activity, adapter, enzyme, or transcription factor activity. Remember, kinase was one of the enzyme which we understood, and the complementary of this was the phosphatase activity. But this all happened after the receptor was activated, and one of the first event for receptor activation was the ligand binding. So let's see how different receptors were activated, whether it was the receptor histidine kinase, receptor-like kinase, receptor tyrosine kinase, or integrins. We saw that there was a dimerization event which induced the activation of the receptor. In case particularly of receptor tyrosine kinase, we saw how the internal regions or cytoplasmic component of the receptor are engaged in regulating the autophosphorylation and dimerization event. In some cases, particularly which were example of receptor serine threonine kinase in animals, the BMP, bone morphogenetic protein, or TGF, tumor growth factor, or activin kind of receptor, they formed heterotetramer. In this particular case, we saw a receptor of two types. If it is receptor type 2, and there is a receptor which is type 1. Each one of them were present in duplicates, and this one had the enzymatic activity. And this had the phosphorylation site. And this was one of the earliest event for receptor activation. So whether it was dimerization or tetramerization, heterotetramerization. That was the basic need for activation. We also saw in case of structures which were formed in case of EPH efferent signaling, where they formed oligomeric clusters. In this scenario, we saw how the two cells, one which is expressing the ligand and the other one which was expressing the receptor, because of this interaction, there were further oligomeric structure forming, same type of receptor coming, and they are making the oligomeric structure. This helped in the process of active signaling, which was leading to the repulsion. And we basically made a oligomeric signaling cluster. In here we saw how the receptor and the ligand could be on the same cell. Further, we saw that some receptors, they actually did not have any activity, but their dimerization brought together the kinase or the enzymatic activity which they were associating. So in this case, we saw that JAK-like kinase, they were brought together. In this case, what we saw, that the receptor to which JAK was binding, that is dimeric in nature. This brings the two JAK molecules together, initiate cross phosphorylation event, which will phosphorylate the receptor also and ensure that signaling continues. Further, we noticed in case of plants that some heterotrimeric structures were being formed. Over there were simple receptors of two different types. Suppose there was something called BAC1 or somatic embryogenesis related kinase. They are forming the heterodimer. On top of that, you have this RLP, receptor like protein, which was coming together to ensure that signaling moves forward. So what we understand, that signaling initiation required receptor dimerization or multimerization, and from there on things moved inside the cell in terms of meeting the response to the signal. But if the signaling continues unchecked, if it is unregulated or dysregulated, it can really cause problems. One of the simplest thing which it can do is just rapid growth of cells in form of a tumor. And we noticed that how one of the receptor tyrosine kinase pathway was merging onto the RAS, and this RAS basically worked on the MAP kinase pathway. This will give growth and proliferation. Basically, what is going wrong here is that the signaling continues and you have more and more cell proliferation going on. So in many cases, we saw, particularly with respect to the EPH efferent receptor, that how EPH receptor was mutated at multiple residues to ensure that signaling was continuing. People have also identified different mutants of this GTPase, which does not require RTK for activation and thus can cause cancer. So this is also a sort of dysregulation where the receptor and ligand combination is not required. Mutation actually creates a problem. So basically this signaling is dysregulated in this context because of the mutation. It can also lead to the pathological condition. If you recollect, when we were discussing EPH efferent signaling, it was important for cell boundary formation, tissue boundary formation. how it was important for vasculature. And also, if you recollect, in the intestinal villi, different regions of the villi, their distinct identity was being maintained by proper signaling of this EPH-afferent type signaling. If that goes wrong, it will create a pathological condition where you have something which we saw as adenoma. If the signaling continues, it can cause metabolic disorder. We just saw that how GPCR signaling, which is responding to multiple of these small molecules, hormones, peptides, amines and so on, will be important, and we saw how G-alpha 12-13 dependent signaling, where the fatty acid metabolism that was being regulated by G-alpha 12 and G-alpha 13. So if your signaling is dysregulated, it will cause the metabolic disorder accordingly. Further, not to be left out, the developmental disorder, because many of these signaling events which we saw, particularly when it came to the RSTK signaling which was there, or EPH-afferent signaling, they were important for developmental aspects regulation. So if something goes wrong with the signaling, as well as if you recollect when we discussed the notch delta signaling, how critical it was for the neuron specification, or the example we saw for Drosophila sheath and socket cells. So basically all of this will be affected if the signaling which is regulating the developmental aspect is affected. Along with the metabolic disorder and developmental disorder, there could be problems which are related to immunity. We saw how JAK-STAT pathway was critical for interferon, interleukin and cytokine signaling. We also saw that how RSTK type signaling, where activin was involved, it was important for cancer, it was important for allergy, it was important for infection and so on. So if these signaling are dysregulated or unregulated, they will create problem. So what exactly is required in case, or what is there already inbuilt in the system, is the receptor inactivation. And there are multiple different ways by which inactivation can be performed. If you recollect that this activation in many cases was dependent on phosphorylation. So if you induce dephosphorylation, that will be regulating the signal. So inbuilt there are molecules like for phosphotyrosine, there are molecules which will be phosphatases, specific phosphatases which will remove this phosphotyrosine and thus inhibit the signaling going on. It's not just for the phosphotyrosine, rather many of the molecules which we saw, many of the receptors which we saw, they had this regulatory mechanism where phosphate which was added could be removed to make them inactive. We also saw the cases where there were inhibitory interaction of the receptor. So if you recollect the activin type receptor, the receptor heterotetrameric receptor bound to inhibitor, and thus it was kept in inactive state. Only when there is a signal and this inhibitor is removed, you will see the signaling going on. So basically you are ensuring that multiple different ways by which this receptor is inactivated. So if inhibitor was present, their signaling was not happening. post signaling, this inhibitor will be coming and binding and keeping it into the inactive state. Also in case of RSTK of BMP signaling, we saw the canonical signaling where there were proteins which were binding to the receptor, and thus the SMAD which needed to bind, the R SMAD which needed to bind and get phosphorylated, this was not happening. So availability of this receptor inhibiting protein is another way to ensure that a receptor is inactivated and signaling ends. More importantly, you can have this receptor interactor stopping from activated to bind. This is what I ended up telling in the previous slide, where RSTK is bound with this I SMAD, and thus the R SMAD cannot go and bind and get phosphorylated, because this phosphorylation event was very important for its dimerization and its binding with the co-SMAD, and this entire complex was going into the nucleus. So as long as I SMAD is available, this process cannot be activated. So you can think that when signaling has to stop, inactivation has to happen, the level of I SMAD will go or the level of R SMAD will go down, and this binding will be inhibiting the activation of the receptor. We have already seen that the ubiquitination can also happen for the receptor. This example we saw particularly in case of JAK-STAT signaling, where one of the product which was being made was this molecule called SOCS, suppressor of cytokine signaling. What it did over there, that it actually collaborated with a E3 ligase which phosphorylated the receptor. It collaborated with E3 ligase What we saw with JAK-STAT signaling is that SOX was one of the molecule which was made, and that is called suppressor of cytokine signaling. This collaborated with E3 ligase and ubiquitinated the receptor, and thus it was degraded and signaling was stopped. Further, we can think about switching of the receptor from kinase to phosphatase. This particular example we saw in case of two-component system, hybrid two-component system in microbes, particularly in case of Vibrio harvii, where the chemiluminescence was under the study, that when normally it is in the kinase mode, there is no chemiluminescence. When it switches back to the phosphatase mode, then there is a chemiluminescence which is seen over there. So switching between one enzymatic mode to the other enzymatic mode is also a way to inactivate the receptor. When basically you can also do transcription, the signal which is activated ends up transcribing the inhibitory molecule. And this inhibitory molecule will go and bind to the receptor or to the kinase or any adapter effector which is over there. So this is how the receptor is inactivated. This is where the negative feedback will also come into effect, that if signaling was happening, and cyclic AMP suppose was being formed, then PK was activated, which phosphorylated CREB, and the transcription continued. This could serve sending a negative feedback to the receptor for inactivation. So sensing the level of the molecules and sending the feedback accordingly is also a way to inhibit the receptor from its activity. We saw in case of notch delta and EPH-effrin signaling that how the receptor, which is here in case of notch delta, it was getting cleaved so that this entire complex can be taken into the delta or the ligand bearing cell, and this signaling will be further discontinuing because no more notch and delta can be engaged. Similarly, we saw for EPH-effrin signaling that proteolytic cleavage which ensured that the complex is taken into the compartment away from the junction where they are engaged in interaction. And this is where, in case particularly of EPH and ephrin, we notice this transcytosis where this big clusters of ligand or the receptor, they were generating pull-push force, which was basically a fruitful engagement and signaling. This must be culminated into eventual endocytosis of this entire complex into one of the cells. Thus, removing this fruitful engagement at the cell surface and rendering the entire process inactive. What we saw in earlier class, maybe the previous class, that how receptor phosphorylation was inducing the desensitization as one of the events. So here the receptor was receptor tyrosine, here the receptor was GPCR type. The phosphorylation which happened on these residue allowed binding of arrestin, and phosphorylation of arrestin by beta arrestin kinase BARC ensured that desensitization happened and G alpha can no more activate the effector. Following this, what we notice that the receptor is internalized, downregulated by virtue of ubiquitination, which was seen on the arrestins. This helped in the removal of this entire complex from the cell surface into the vesicle, and that's where you realize that the signal is, this is where what we understand, the internalization which happened here allowed removal of the active receptor and recycling of these components, whether it is receptor, G protein or arrestin, to get engaged into the next round of activation cycle. So what we have learnt in this class today, that while receptor activation is important for signaling, it can be achieved by multimer formation in the simplest form, that two receptors come together or more than two receptors come together in different combination, homomeric or heteromeric combinations. The effector or the second messenger activity regulation is very important. So basically receptor is activated and that activates the next set of molecule, their regulation becomes very important. If they are not controlled, it could be a problem. This is where we understood that how RAS mutant can create havoc. Receptors are inactivated. Why? Because you need to stop the signaling, and this will help maintain the homeostasis. If receptor is not inactivated, it will continue to signal. It will create a problem, particularly when it comes to growth and proliferation, because that could lead to the cancerous conditions. And what we have understood as multiple different ways of receptor inactivity is dephosphorylation, inhibitory interactions, proteolytic cleavage of the receptor, ubiquitination, thus internalization, and then desensitization and downregulation. By these different means, one achieved the receptor inactivation and thus culmination of the signaling. Thank you.